We're going to discuss the University of Michigan initiative, but we have Jeff Sturm and Dr. Angel Chin. So give them a round of applause.
Good morning, everyone. It's my great pleasure and privilege to be here with Dr. Angel Chin that I've worked with extensively. I'm also on the advisory board of the University of Michigan ALK Research Initiative, and I get to see all the cool science there, which is, I can't talk about any of it, but it really is a great pleasure. We're also going to, Angel's going to do a presentation, and I'm going to do a bit of a Q&A. And I'm winging that, so we'll see how we do. Dr. Angel Chin earned her MD from Case Western Reserve University, where she subsequently completed a residency in internal medicine. She completed her fellowship in hematology oncology at the University of Michigan and is now a clinical assistant professor at the University of Michigan Rogel Cancer Center. Her research interests include mechanisms of resistance to immunotherapy and targeted therapy. She is the clinical lead of the Judy Tam ALK non-small cell lung cancer research initiative, focusing on ALK-positive NSCLC patients that have become resistant to TKIs. She's an active clinical investigator with multiple investigator-initiated trials and participates in cooperative group and industry-sponsored trials. She is a member of the SWOG. What does that stand for? Southwest Oncology Group Lung Committee and NCCN Small Cell Lung Cancer Committee. What's that?
National Comprehensive Cancer Network.
No, National Comprehensive Cancer Network. She is the co-chair of the Thoracic Oncology Clinical Research Team at the University of Michigan. So let's have a little hand for Angel Chin.
Good morning. Thank you all so much for having me here. It's been such a wonderful 24 hours in Philly, and I'm just inspired by everybody that I've met here so far, and I look forward to working together in the years to come. So the title of my talk, given to me by Ken, was Defining the Molecular Aspects of ALK-Positive Non-Small Cell Lung Cancer to Find New Treatments. So for those of you who may or may not know, we have something called the Judith Tam ALK Lung Cancer Research Initiative at the University of Michigan. That's a picture of Judy. She is a patient, was diagnosed with ALK-positive non-small cell lung cancer probably about 1.5 years or 2 years now. And she is a great philanthropist, and she was so charged after learning about her diagnosis that she donated a large sum of money to us to really push forward research in this area. Our overall lead is Dr. Mariver, who is an established scientist and has a long track of research. And together we have this large team to really focus on ALK-positive non-small cell lung cancer. So we have 3 main projects in our initiative that are currently ongoing. And I'll say that this list is constantly evolving. So we want to advance precision medicine by testing minimally cultured patient samples. So samples that patients give to us to help us predict response to therapy. We want to develop models to understand tumor cell of origin events that lead to resistance. Because I think the question that I certainly don't know the answer to, and I don't know who does, is what is causing ALK-positive non-small cell lung cancer? Yes, we know all about the fusion, but why do you get the fusion? I don't know the answer to that, but I think that's an important question to understand. And then we also are working with a medicinal chemist to create different types of ALK inhibitors, very similar to some of the different things that Dr. Shaw mentioned in her talk yesterday. We also have 2 developing projects that are ongoing. So we want to study the role of circulating tumor cells in monitoring response and development and resistance. I think this is interesting. We all hear about liquid biopsies, but usually liquid biopsies, at least currently, is looking at something called circulating tumor DNA. So this is DNA that is in the blood that is from cancer cells that have kind of lysed. what we think may be maybe complementary or even more interesting is actually capturing the actual circulating tumor cells. So these are the cells that are swimming in the body and potentially could be causing problems. We want to be able to study those and understand, in real time what that cancer cell is doing. And then our second developing project is to understand the tumor microenvironment and the interaction within the immune system. I think we've also heard, that conventional checkpoint inhibitors, PD-1, PD-L1, doesn't seem to be very effective for ALK non-small cell lung cancer. Why is that? What is going on with the tumor microenvironment? What are your immune cells doing with your cancer cells, and why is this not working? And I think we'll talk a little bit more about that later on with Jeff when we talk about the Memgen collaboration in that study. So a little bit about, how we evaluate drug therapy efficacy and resistance. So this is where all of your participation is so important to what we do. So if there's any kind of procedure, biopsy, fluid, whatever it is that you may be undergoing and you're interested, we would love to be able to collect that tissue or sample. We then are going to be able to take the sample and start collecting circulating tumor cells, the sample itself. We will be asking you for some pretty detailed history in terms of what you've gone through, what your sequencing information is, what your response has been in terms of clinically with your prior therapies. We also have a dedicated pathology team that is just sequencing for this ALK project. And then from that sample, again, whether this is fluid or tumor, that is where we do that drug testing. And, there are a lot of different places that do drug testing. This is not I think, a novel concept in that sense, but I think what we try to do is we try to curate that panel of drugs to each individual patient, right? We're not going to give every single patient the same drugs because it doesn't make sense. We want it to be personalized. And then we can kind of get a sense of what these drug curves would look like. And I'll give you an example of that as well. We're also going to try to generate what we call patient-derived xenograft models. So these are called PDXs. So essentially allowing that tumor to propagate in mice. And this is really important because we know that it's hard to come by adequate tumor tissue to be able to do all the studies that you want. And you want to be able to study the evolution of how these cells behave. And so one of the ways we can do that is by establishing these PDX models. And then we can do clinical trials on it, right? We can take mice that has patient tumor and we can see what is going to work in this mouse. Not that saying that what works in the mouse works with us, but it is a start. And that is too where our medicinal chemists can come in and kind of test some of their new ALK inhibitors. So I want to give an example of results because I think this is something that's a topic of interest. So this is, I will just say very briefly, this is a patient who was able to donate a sample to us. It was fluid, it's pleural fluid. And this patient had progressed on alectinib and lorlatinib as patients do. When we're looking at this curve, what we want to focus on is that column that says Cmax. What a Cmax is the kind of the maximum concentration of drug that can occur in a certain compartment of your body or a certain part of the body before you administer the next dose. So what is the highest level of drug before it kind of gets broken down and processed out? I will say that the Cmax for a lot of oral drugs are known. That is part of the testing that occurs when we test new drugs in patients, as we find out what the Cmax is. I will say that sometimes it's harder to predict what that is for IV chemo drugs, right? Because these drugs are now given as an IV and it's hard to know what that maximum dose truly is. When we look at this chart, I'm a simple person. Green to me is good, red is bad, and that's how it plays out here. So green means that these cells are very sensitive to the drug, meaning you're getting good tumor cell killing. Red is bad. Red means that these drugs, even though, these tumor cells, even though you're hitting it with the maximum concentration of drug, they are still hanging out and they're alive. So if you look at this, this patient sample, you'll see kind of towards the middle bottom, alectinib, and even towards the more bottom, lorlatinib. So we kind of did this as a proof of concept that yes, the tumor cells that we got and tested reflect this patient's history, and that the alectinib and red are very much, lorlatinib, are very much on the red spectrum, so bad. So that is why going up then, now we're starting to look at drugs that are green, and brigatinib pops up on there, repotrectinib pops up on there. So it's just some food for thought. But I think one of the things that Jeff and I have really talked about, as well as with other committees, is, I think research is obviously very important, but we need to think about what we can do right now, right? We want to advance science for 5-10 years down the road, but there are patients that need our help right now. And so what can we do about that? I will not discuss this again because Dr. Shaw obviously wrote this paper and so gave a great talk about this yesterday in her presentation. But again, just to remind everyone that there are different mechanisms of resistance to ALK, whether that's on target, whether or not that's a single mutation or something called compound mutations, whether or not there's these other pathways that are active. And this is by no means a comprehensive list of all the different pathways that can be activated, or on that far right, when the type of lung cancer changes. So these are things that we all have to come into mind for each individual patient that comes to our clinic that have progression. I really like this paper that was published from Dr. Aaron Hada's group out of MGH last year. And this is kind of one of our biggest samples of 48 patients who had progression after lorlatinib. And Dr. Hada and his group did an extensive set of tests and to see what is happening. So you'll see that about half these patients, they did not detect that mutation anymore. So is this the off-target resistance that we're talking about? About 20% of these patients develop a single mutation, and about 30% of these patients developed compound mutations, which is consistent with what we see kind of when patients progress on a third generation ALK TKI. So if I'm sitting in the clinic and I have this information in front of me, the question I'm going to ask myself as well, what treatment options do we have? So if we have no mutation, am I going to give them chemo, clinical trial? Is there still a benefit of continuing their ALK TKI? If it's a single mutation, think about, again, clinical trials or the next generation ALK inhibitors. Do I now add chemo to their ALK TKI? And then when we have compound mutations, it truly is a grab bag because all compound mutations are not created equal. So some compound mutations, as you can see, again, green is good, red is bad, yellow is in between. Some compound mutations, you'll see that may be a little bit more resistant to alectinib or lorlatinib. You'll see that actually may convey sensitivity to an earlier generation ALK TKI like crizotinib or even brigatinib. But there's some compound mutations, and some of them are triplicates, as you see in the bottom, that pretty much convey resistance to every single current ALK TKI. And so I will say that even when a patient has a compound mutation, the answer to what comes next is not the same for every single person. And again, I think this highlights the importance of individualized care. So this is kind of my wish list for what would be an ideal post-lorlatinib TKI. Obviously, I think we need to have activity against single ALK mutations. We have activity against compound ALK mutations, and the ones that include G1202R and I1171N are kind of the main culprits that pair with another ALK mutation. I would love it too if they also have some activity against ALK independent pathways. Of course, I think on this list, we need to have things like CNS penetrance, tolerability. I think my wish list could be really long, but I think these are some of the ideal features. So this is the clinical trial that we are working really hard to open at University of Michigan. It is a phase 1 study of a drug called gilteritinib for ALK-positive non-small cell lung cancer. Now, gilteritinib, thankfully, is something that's already approved by the FDA, but it is not in lung cancer. In fact, it is approved by the FDA for a type of leukemia called FLT3 mutated acute myeloid leukemia. The reason that we are interested in this compound is that gilteritinib is something called a multi-kinase inhibitor. You can market it to target FLT3, which is what it's currently marketed at, but it has activity against many of these other kinase pathways, which includes ALK, but also some of these pathways like ROS, RET, AXL, some of these things that we're seeing as a mechanism of off-target resistance, this drug also happens to target. This Japanese group here has already done some very interesting work on lung cancer cell lines. They're comparing the 3 drugs, alectinib, lorlatinib, gilteritinib, and they're looking at something called the IC50 value. Now, an IC50 value is like a pharmacodynamic way of explaining how potent a drug is. It means what is the concentration of drug that is needed to kill 50% of the tumor cells. And you want something with a low IC50 value. That means your drug is really potent. And so if you look on the graphs, you'll see that gilteritinib has a lower IC50 than the other 2 drugs in single ALK mutations and in compound ALK mutations, and even some of the more resistant compound mutations. There's encouraging activity. And then since we conceived the study, have also been doing this in our patient samples. This is from a different patient than I showed you earlier. This patient had progressed on alectinib, lorlatinib, and also the combination of carboplatin pemetrexed. And that's why I had mentioned earlier that the Cmax of IV drugs is hard to interpret, because as you'll see here, the top drug here that works is carboplatin, but we know that's not true. So, but if you look at alectinib and lorlatinib, again, they fall very far down on our chart, very red, but gilteritinib is once again in the green. So at least in the patient's tumor cells that we have, it suggests that there is maybe a benefit of gilteritinib. So where we are on the study, so thanks to all of you, it's been funded through a grant that's with ALK-positive longevity. The makers of this drug is Astellas. They have agreed to provide drug for this study. So kudos to them. I have written the protocol. It is in the regulatory process because this drug, even though it is FDA approved, we're trying to use it in a very different indication than the FDA approval. So there is still some additional paperwork that needs to be done, but we are really, really hoping that before 2024 starts that we can open this trial. So what's next? I think thanks to Jeff and Ken, I have to correct myself. I think it's important to say that ALK non-small cell lung cancer had been considered rare, but I think sitting in front of all of you today and all of you who are at home, I think that's perhaps a misclassification, but it is impossible, as we all have seen this weekend, to do any studies without our collaborators, without our patients, without our care team. And because I am currently at University of Michigan, I do have to quote a very famous football coach. His name is Bo Schembechler. I am not from U of M, so like my husband had to tell me this. And he just said something called the team, the team, the team. And I don't think there's any better model for a football team and the ALK-positive team than that quote from Bo. I think it's important that we can learn something from each individual. I think patients have come up to me and say, hey, I haven't progressed yet. So like maybe, you don't need my sample. That is not true. I think we can still learn something from every single person. Jeff and I will talk a little bit about this more as well, but there's something called a materials transfer agreement, which is the bane of all our existence, to be able to receive some of these patient materials. But we are actually working on getting an IRB protocol to exempt that when it's only blood, and to be able to give the patient a kit that they can take to their blood and then send it back to us directly. One of the other big things that have come up as a point of contention is sharing those drug testing results. So I showed you those pretty graphs. I guess, we would love to be able to share that result with the patient and with their oncologist. The reality of this, though, is that it is still research, right? So we never want to be able to do this and say, this is a prescription for what you should have next. We want this information to be additional extra piece that your oncologist and you and your care team can look at and say, okay, do these choices make sense to us? But thankfully, we're actually making progress on that. We actually convened an ethicist, a bioethicist onto our team, who is going to help us trying to maneuver the delicacies of delivering these testing results. So I'm really excited to be able to do that. Speaking of the team, this is just a list of all the team members at U of M working on this initiative. There's lots of us, and we add more. These are our current MTAs, or material transfer agreements, that we have with different institutions. So we have active, we have things that are under negotiation with the lawyers, and then we have places that we've just started to have some discussions with. And we hope to continue to build on this list of collaborators. And this is just a bit of an update of so far what we've been able to collect, both from ALK-positive non-small cell lung cancer patients and also from patients who have a diagnosis of lung cancer in general. Because I think some of the same research and experiments that we're doing in ALK non-small cell lung cancer is something that can really set the standard for other types of non-small cell lung cancer as well. That is our email. This is our clinical team. You should feel free to email us at any time. And we look forward to hearing from each and every single one of you. So thank you.
Hello. So the team that you brought together since really only the beginning of 2022 was from all over the U-Michigan teaching institution. Can you talk a little bit about the level of those people and the experience depth that they have?
Yeah, so our initiative is young since the beginning of 2022, but I think there's a lot of passionate people who do a lot of really cool things that are all really excited by this cause. I think a lot of our collaborators, when we first approached them, I don't think they really knew what ALK non-small cell lung cancer was. They're like, oh, isn't it just like lung cancer? And then when they heard more about what was going on and the different treatment options, they're like, oh my gosh, this is such a different concept of lung cancer. But, we have PhDs at the highest levels doing science I can't explain. I just sit there and nod my head. They're dissecting down things to the molecular level. We have molecular pathologists, we have chemists, we have bioinformaticists who are taking this wealth of data that we generate, and they're going to try to parse it out down to, okay, what is actually clinically relevant, right? Because I think as the clinician, I'm sitting there, I'm like, what do you want me to say to my patient? What kind of information can you give me to make this make sense for a patient? And I think having that manpower in our team has just been so incredibly wonderful to see.
So yes, no, okay. We've called on the patient group to donate tissue to the University of Michigan, and it hasn't been that easy. The material transfer agreement between the patient's clinic and the University of Michigan has proven a bit difficult to arrange. Can you tell us why we need that material transfer agreement?
Yes, so this is something I learned during this initiative, because I think, like many people, when I started, I was like, oh yeah, I mean, if a patient consents, why can't we get their tissue? And the issue is really more, it's a legal issue, legal matter, in the sense that when a patient signs consent to have a biopsy, to have a surgery, they're signing consent that that tissue now belongs to the institution. The institution often will have biorepositories themselves to be able to do research. The institutions have obviously a vested interest in keeping that for research purposes. So it's not so much then a patient consenting, it's now the institutions have to have this contract, an agreement, an MTA, as we call it, to be able to share that that precious tumor sample amongst each other, and all the legal implications of who actually owns it, and when you generate data from it, what are the what can we do in terms of publications, collaborations, things like that, is a big red tape and hurdle that it is very difficult to overcome, but we have overcome, as we have seen in the list of the different institutions, but it takes time and perseverance on all our parts.
Let me amplify that from a patient's point of view. When you're sitting there in the pre-op room, perhaps with an IV in your arm, or you've at least taken one shot, they hand you a bunch of paperwork. Usually it's a multi-part form, and somewhere in that multi-part form, you give away all the rights to your tissue. It's not yours anymore. You don't even know it. Whoever reads that stuff, right? So in order for the University of Michigan to acquire your tissue in a way that they can actually use, there has to be a material transfer agreement arranged. And those lawyers, they might not even work in the hospital. They might be outside attorneys in your clinic. So it's really difficult. It's a difficult process for your doctor to manage through 5 or 6 layers in the clinic of bureaucracy to really accomplish a material transfer agreement. And they say that's just too difficult to do for the most part. But the issue is one of custody of the tissue, legal custody of the tissue, and the integrity of the legal custody of the tissue. So from start to finish, University of Michigan has to have legal custody to do whatever they want with your tissue. And that process, overriding the existing process, is not easy, as we've shown. My hospital wouldn't do it. They wouldn't even talk about it in Northern California. But University of California, San Francisco has managed the process and has a material transfer agreement. They're in my backyard. I'm already a patient there for something else. So I'm transferring my records over there. And I would suggest that people that really want to do it is to find one of the hospitals, MGH, for example. A lot of us have gone there and continue to go there, has a material transfer agreement now with the University of Michigan. So if you're really bent on doing it, you could do it at one of the already approved and growing list, or perhaps go to University of Michigan to donate your tissue in the right time and the right way. You have to sign up for the program earlier. My detailed description of how to do that is on the Facebook site in the documents section, and I'll publish it again early next week so that you can contribute. And now I'll hand it back. Do we want to talk about MemGen?
Yeah, so one of the things I think ALK-Positive has been so incredibly instrumental to our research initiative is connecting us with other companies, with pharmaceuticals, with biotech, who are developing some pretty cool new treatment options, and maybe haven't thought about ALK non-small cell lung cancer, but now they are, and putting us in contact with them to maybe be able to study their compounds that are even in clinical trials, or thinking about using their compounds for ALK-positive non-small cell lung cancer. So one of the exciting collaborations that have resulted to date is with a company called Memgen. They have developed something called an oncolytic virus. So sounds scary, very futuristic, but it is basically redesigning a virus to do what we want it to do, which is help us fight cancer instead of causing us illness. Now, what's I think really interesting about the concept of oncolytic virus is, as you imagine, when you get sick from a viral infection, you have this very systemic immune response to it, right? And as we've said before, there's something about ALK non-small cell lung cancer that currently does not allow our conventional checkpoint inhibitors to be very effective. But is that something that we can do? Can we somehow reprogram the immune system with something like an oncolytic virus? I think that's an area of interest. So right now, one of our collaborators, Dr. Ligasetti, he is a surgeon, but he also is an immunologist and has his own lab. They're going to start beginning experiments with the Memgen oncolytic virus, MEM288. And the experiments, as he has explained it to me, is that they're going to be, again, taking these mouse models where they have not tumors that are kind of growing underneath the flank. They're going to then start doing experiments to inject the oncolytic virus into these tumor models. They're going to do that with and without alectinib, so both by themselves and in combination with alectinib. As you can imagine, when you are doing testing in terms of injections, the interesting part of this is that you can then take that tumor out of the mouse and you can break it down by all the different immune cells that are there. You can take the blood of the mouse. You can see what is happening, what is changing in terms of the population of the immune cells. You can take lymph nodes around there, right? And see, okay, so what is now the lymph or the immune system responding to this? And so I think that's a really exciting area. And I think much like Dr. Awad's study at Dana-Farber, where they're looking at potentially an ALK vaccine, I think this is a similar concept of somehow reprogram the immune system to help work for us in ALK.
So the Chief Science Officer of Memgen is here today. Dr. Mark, you want to stand up and say hi to everybody? You in the room? Thank you so much for joining us. So Memgen is one of the prototype successes of the ALK Research Acceleration Committee, where we're bringing together the patients. So the tissue that's going to be injected into these mice comes from ALK patients. Probably one or two of those mice will be injected with ALK-positive patients, members. The money's coming from ALK to run. No, is that right? No, that's not right. That's gilteritinib. Part of the money is coming from ALK to run that trial, that experiment, and certainly University of Michigan. So you've got biotech, academia, and patient group collaborating together in every way to make something happen. And we hope to see a whole lot more of that down the road. And oncolytic virus, they inject the oncolytic virus into your tumor. And in some of the preclinical experiments, it not only kills that tumor, but it goes around your body and kills tumors elsewhere. Abscopal effect. The scientists come up with all these crazy names for things. It's abscopal. I mean, how about at a distance, So one of the focuses that Angel mentioned is to help patients right now. So if you can get your tissue through the material transfer agreement to the University of Michigan, they're going to test it and against a panel of drugs and see which one works. Now, at some point, Not right now, because there's some registration issues. You're going to be able to provide the results of those tests to the patient's oncologist. And that's kind of the holy grail of drug testing specific for a patient. Could you elaborate on that?
Yeah, so Dr. Mariver has actually been visiting a number of other big cancer centers and kind of publicizing our work. Again, just this is part of the team, right? We need as many collaborators as we can. And when she showed her results, what she told me was that the response was so strong from some of these cancer centers that we must be able to share these results. That is why we've accelerated our attempts to be able to share these results in the exact same format that you just saw me present and with greater detail. And that is why we've brought on a bioethicist. We are trying to advance our ability to have something called CLIA certification. So CLIA certification is basically when a national governing body for pathology has said that your assay, your test meets the standards to be able to be used in a clinical setting. Again, I don't think we ever want to say that our drug testing is going to be a prescription, right? It's not saying I'm going to prescribe these drugs in this order. I think though what this drug testing does in terms of when I've experienced it is it really helps me understand what makes sense. So if a patient's progressed on alectinib and lorlatinib, I'm now taking their clinical history, their sequencing history, and I'm like, I feel like I need to add blank. And I'm looking at my drug testing results. I'm like, yes, I already have some proof that this combination is worthwhile. I think that is where the benefit of that comes, right? And then it will maybe allow patients to think about clinical trials and may that information help them decide what kind of clinical trial are we going to look at? Chemotherapy, are we going to look at something else may be beneficial. So absolutely, we're working really hard to be able to share those results with the patients and their oncologists.
Thanks. So gilteritinib is another tripartite success between Astellas, a pharmaceutical company, and University of Michigan, an academic institution, and ALK-Positive. We helped facilitate the donation of that drug from Astellas. Shout out to Julie Silberger in the virtual world, wherever you are. It was part of the team that did that. Who is gilteritinib going to be good for? At what point in a patient's treatment plan would gilteritinib come into play?
Yeah, so right now, the way we've designed the study is something called a phase 1. So for those of you who may not be well-versed in clinical trials, there's different phases of clinical trials, and each phase is looking at a different endpoint. So phase 1 is when you're taking a compound, either completely new or doesn't have yet data in your disease, and we're really looking at safety and tolerability, right? I think before we start giving this drug to a large number of patients, we need to know that it's safe in this patient population. And with a lot of phase 1s now too, we're also generating response information. So how well is a patient responding? That usually will be able to generate data to support a phase 2 study, where now you're looking really, your primary endpoint is efficacy, and you'd be able to enroll enough patients to test that efficacy. And then the holy grail of clinical trials is something called a phase 3 study, where you're taking your intervention and comparing it to what is current gold standard, and you randomize it head to head. And if your intervention wins in terms of overall survival or whatever marker is meaningful to you, that is when you consider your drug to be a success. So the gilteritinib study, we designed it in mind for what the ALK population needs right now. So the only stipulation, if you will, is that you, patient, must have progressed on lorlatinib. That is it. You can have had any other number of treatments, whether or not you've participated in other clinical trials, whether or not you've gotten chemo, whether or not you've gotten other TKIs. That's all okay, because again, when we're thinking about designing a clinical trial, we think about what the patients need now. And we don't want to exclude any patient, really. The only reason we say you have to progress on lorlatinib is because that's the standard of care. We're not ready to say that our drug is better than lorlatinib. I don't think we will be able to say that for a long, long time to come. So we don't want patients to not get lorlatinib and instead do something experimental. But if you've progressed on lorlatinib and you're interested, this is where our trial is.
So the original paper that came out of Dr. Kawakami's lab that stimulated, I think, your interest, that ALK-positive became aware of very quickly after it was published. Dr. Kawakami is a Japanese ALK researcher that worked in the labs at Massachusetts General Hospital for a while, and all the people over there know him quite well. And he published a paper that said gilteritinib was active against a lot of the mutations for ALK. His original paper showed efficacy in combination with lorlatinib, and you're testing initially in non-small cell lung cancer patients as a monotherapy. What's the rationale for that?
Yeah, so I think that's a great question. I think when we're trying to test a new drug, what the fear is that if we test it in combination, so first of all, we don't know really about what the overlapping toxicities are. And so I think we would not want to prematurely say that there is no benefit because there's patients had too many toxicities. And so part of it is not, shooting this down as a potential drug of interest too early. The nice thing about taking or repurposing a drug that's already out there is that there's a lot of information that's already published about the side effects of gilteritinib. And at least from what we can see, fingers crossed, it does not seem to have a lot of overlapping toxicities. But the truth is that patients with acute myeloid leukemia are a very different patient population than patients with ALK non-small cell lung cancer. I think this is just the beginning of what we hope to accomplish with gilteritinib, right? Single agent right now, but also a wonderful picture. But if I think that if, we are able to see that there is an efficacy signal, absolutely, we would love to be able to do it in combination with gilteritinib. But I think we got to take baby steps first, ensuring the safety for all of our patients before we start doing combination.
So a few months ago, I asked Dr. Mariver if we know if gilteritinib is brain penetrant, and you folks have developed a unique method for determining whether a drug is penetrating the brain. Could you discuss that a bit?
Yeah, so one of the obviously one of the big concerns that we all have is when patients develop brain metastases, and one of the things that we enjoy with our TKIs is their CNS penetrance. When we look at studies of gilteritinib now, not much is written about their CNS penetrance because with acute myeloid leukemia, it's not very common that patients have CNS disease, but there's actually been a few published reports of patients who had CNS penetrance for the leukemia on gilteritinib who actually had benefit to gilteritinib in the CNS. At the same time, Dr. Mariver and other super smart people are developing this mouse model where we're able to administer gilteritinib and then kind of assess the level of gilteritinib that is available in the mice in their central cerebrospinal fluid. So we're working on those experiments.
So I'm currently progressing on G1202R and lorlatinib with two unique mutations, I1171S and G1269A, and it took me a month to memorize those, I might add. And fortunately, it looks like gilteritinib has strong response for all three of those mutations. Want to comment on that?
Well, when the study opens, I have your email, Jeff.
With that, I'll say thank you very much. I have to say, I've had the great opportunity to work with all the people at the University of Michigan. They are the finest people I've ever run across, and they're they're doctors, and their heart is really in it. And it's not just the technology, and they care about what they're doing very, very deeply. So let's have a big hand for the Angel Chin and the people at University of Michigan.